Hello, everyone. It's fantastic to be here and show a little bit what we've been doing with long COVID in a bunch of the different biobanks that have either EHR or symptomatic recording of long COVID. So my background is actually in sleep disorders, and I've particularly been working on an infection-triggered sleep disorder, narcolepsy. And infections can be indeed risk factors for many later diseases, like chickenpox and shingles, and influenza, and especially pandemics in the onset of narcolepsy. And I do a lot of genetic studies, and they have been extremely powerful for understanding some of the biological mechanisms that contribute to later sequelae of the original infections. For example, in narcolepsy, in the 80s still, people thought that it's primarily a neuropsychiatric disease because of the intense fatigue and hypnagogic hallucinations that the people were experiencing. But when HLA typing results became available, it was pretty evident that narcolepsy was actually an autoimmune disease and triggered by infections, particularly with influenza A. And we see this also with genetic studies of narcolepsy, where a lot of the genetic risk factors overlap with influenza infection. So when COVID pandemic began, we started a consortia to put together many of the different cohorts that have information about long COVID. And one of the efforts that was coordinated from here in Boston and from University of Helsinki, that are my two affiliations, was to understand why some individuals have such strong differences in response to infections, some being essentially asymptomatic, others developing severe disease or even dying. And of course, we were interested in the recovery. And as part of this work, I also want to say that this COVID host genetics initiative was extremely powerful for understanding biological factors that contributed to either infection susceptibility or severe disease. But also now when we have done a follow-up of this study and looked at Omicron variants, we see quite a big difference in the biological mechanisms that contribute to earlier viral strains versus the later viral strains. So if you want to read our preprint on the glycosylation differences and host factors on Omicron versus Alpha variant, that's in press and it's pre-printed. So we also put all of our summary statistics available if anyone would like to have that for their analysis. And I just want to do like a quick disclaimer. So when we talk about genetic variants, we have a couple of high predictive variants like HLA for a subset of autoimmune disease, or APOE or BRCA1 mutations for breast cancer screening. The type of genetic associations I'm showing today are more informative for biological mechanisms and should not be interpreted as diagnostic biomarkers. So they really tell about what underlying mechanisms contribute to long-term disease rather than being ready for clinical testing at individual level. So we were lucky to get a bunch of different cohorts, so over 30 cohorts for the initial study from 19 different countries. This data was recently published in Nature Genetics in June, and I'm going to first show this and then a few examples of unpublished data. And here we had either long COVID by ICD-10 codes or then self-reported symptoms. And we compared long COVID to either population controls, regardless if we had information about their earlier COVID infection. And this is partially because we don't want to have a collider bias between cases and controls, and partially because not everyone could get tested, so we didn't want to limit the sample size there. And then we also, in the subset of cohorts that had testing available, we also compared with test-verified COVID the genetic associations that we were looking at. And really, like has been shown by many previous talks here, is the overlapping and largest symptom here is fatigue. So as a sleep researcher, I think there is a lot of opportunity also to understand sleep and long COVID, especially because in long COVID, the symptoms are quite different from some other sleep disorders. So in long COVID, we see both insomnia and fatigue, and the key being that the sleep is not very restorative, even if you are able to sleep. And then, of course, we have all of these other symptoms, and we were pretty inclusive in the definition of long COVID and included as much data as we were able to get from the early 2 years of the pandemic. So, how do you do this study? Is that each cohort analyzed their data separately, and then we meta-analyzed it centrally. And this is a nice approach if you ever need to share data, because there is no individual level genetic data transferred between countries, so it's safe to send the results from your in-house analysis. So we had 3 data analysts working on the meta-analysis of this project. And I'm gonna show you how to read this plot. So each individual point here is one genetic marker. And on the x-axis, we have the human chromosomes, starting from the very first base pair all the way to the last base pair and to the chromosome 23. And on the y-axis, we have then the significance, and it's in a negative log 10 p-value. So the higher the peak, the more significant the finding. And you start seeing these skyscrapers showing up in the plot because genetic variants close to each other are correlated, and this is called a Manhattan plot. And we found, in this very first study, we found association at the FOXP4 locus in chromosome 6. So FOXP4 is primarily expressed in the lung. It's also expressed in the brown adipose tissue, where it participates in energy metabolism, and it's also expressed in hypothalamus. But the lung data was super interesting because it was quite specific expression in the alveolar macrophages and was also later related to defense against COVID infection itself. And the strongest associations for other phenotypes were a lot of other lung-related phenotypes like severe COVID-19, but also lung cancer. And higher expression of FOXP4 was related, or the same allele was related to higher risk for long COVID. And I just want to mention that this was an international consortia, so it's really important to have data sets across the world because the variant that we discovered in this study is extremely rare in the European population. So we really needed a multi-ethnic genetic ancestry to be able to pick up the signal. Okay, I'm just going to briefly mention that the question I get most often is that is FOXP4 just a signal for severity? And to go show that is that if we plot the effect size of COVID severity or COVID infection susceptibility versus long COVID effect size, we can see that FOXP4 really stands out as specific also for long COVID. And now there is an interesting paradigm, of course, so in order to get long COVID, you need to have contact with COVID itself. So a lot of the variants that are related to COVID infection or COVID severity are also risk factors for long COVID, and like they should be. So finally, I just want to use the last minute and a half that I have is that there are also other GWAS for COVID, and there was a really nice preprint put by 23andMe that discovered HLA, ABO, and BPTF, and then also German study of long COVID. And we have actually now analyzed additional independent data from 23andMe using our questionnaire-based data, and we see all of these 3 associations that were put out by 23andMe, so ABO, HLA, and BPTF, which is also a transcription factor like FOXP4. And the other point I would like to highlight here, that my group also coordinates a large-scale consortia on fibromyalgia, and we also participate and collaborate with Chris Ponting on the DECODE-ME replicating their findings. So for fibromyalgia, this is also pre-printed, and I'm happy to share the summary statistics if anyone wants them, is that we can see the BPTF that was associated with long COVID, along with signal at the HLA, also with fibromyalgia. So there is shared genetic architecture and risk factors across all of these 3 domains, fibromyalgia, ME/CFS and long COVID. So to summarize, FOXP4, HLA, ABO, and BPTF are associated with long COVID and primarily highlight immunity and lung pathophysiology. And it's really important to have these large-scale cohorts. And shared associations are present for fibromyalgia and ME/CFS. And my group coordinates, together with our collaborators, the Long COVID Host Genetics Initiative and the GEM Consortia, which is Genetics and Epidemiology for ME/CFS. We're a pretty fun and collaborative group, so if anyone wants to collaborate or get some of these data, please contact me. Just want to highlight the massive number of people in the Long COVID Host Genetics Initiative and everyone in our team. Thank you so much.